of two carbon compound, we call it 2-phosphoglycolate. This is 2-phosphoglycolate. You see, this is 5 carbon compound. Once it is oxidized, it forms 3 carbon compound, phosphoglyceric acid, and 2 carbon compound, we call it phosphoglycolate. Therefore, this reaction is catalyzed by the enzyme known as Rubisco. Okay, therefore the process in which the Rubisco fixes oxygen to the Rubip to form 3 carbon compound and 2 carbon compound, we call it photorespiration. Okay, photorespiration, as you can see from this word, photo means light. Respiration, as a normal respiration, we know respiration, it uses oxygen, but also it releases a carbon dioxide. Therefore, because this process, it occurs under the presence of light, but it uses oxygen and also it releases carbon dioxide, and that is why it is referred as photorespiration. Therefore, photo means because the presence of light, it occurs in presence of light, but also respiration because it uses oxygen, but again it releases what? A carbon dioxide. That's photorespiration. This photorespiration, actually, it lowers the yield of C3 plant. Or we are saying, when the photorespiration occurs, it lowers the chance of photosynthesis to occur. As you can see here, the formation, the product of this photorespiration is a PGA and phosphoglycolate. This phosphoglycolate cannot enter into a Calvin cycle. Rather, it is converted into different compounds through 3 organelles, which are chloroplasts, peroxisomes, and mitochondria. Okay, therefore we are saying this process is a wasteful process because it lowers the yield of photosynthesis in C3 plant, but again it uses some of the chemical energy produced from the light reaction, for example ATP. Therefore the process, it uses it. It uses ATP, but also we are saying it lowers the chance, or it lowers yield in C3 plant for about up to 50%. 50% rate of photosynthesis is lowered by this photorespiration. Therefore, it is wasteful. It is wasteful process. So the process of photorespiration, it lowers the yield in C3 plant, and that is why we are saying when the photorespiration occurs, it lowers the chance of photosynthesis to occur. And the plant which are more prone to this photorespiration is C3 plant because of the leaf anatomy of the C3 plant. Okay, what are the factors which influence the process of photorespiration to occur? Actually, we are saying there are 2 main factors which influence the process of photorespiration to occur, and these are high temperature and high concentration of oxygen. These are 2 main factors. Okay, the factor number 1 which influences the process of photorespiration is high temperature and factor number 2 is high concentration of oxygen. High oxygen concentration. Okay, how this factor bring about a photorespiration? Actually, we know at a high temperature, for C3 to survive, there must be a regulation of the opening and the closing of the stomata. Therefore, at a high temperature, the C3 plant tend to close, or the stomata of C3 plant close. And once they close, they reduce the amount of carbon dioxide which will diffuses into the plant tissue. And when there is a little amount of carbon dioxide diffuses into the plant tissue, it can result into photorespiration. If you remember, we have said this photorespiration occurs when there is low carbon dioxide concentration. The stomata close, little carbon dioxide diffuses into the plant tissue, and therefore photorespiration may occur. And this high oxygen concentration, actually, when there is high oxygen concentration, if you remember, the C3 plant had the so-called rubisco. This rubisco can fix oxygen and carbon dioxide depending on the concentration. When there is high oxygen concentration, the rubisco fixes oxygen to the rubip, and it results into formation of one compound with 3 carbons and another compound with 2 carbons. Therefore, the high oxygen concentration can favor the process of photorespiration to occur. Okay, let's see how this photorespiration can be explained diagrammatically. Yes, as I said, after formation of this 2 carbon compound, or we call it phosphoglycolate, this phosphoglycolate is converted or is exchanged between different organelles such as chloroplasts, peroxisomes, and mitochondria. Therefore, we have this, the first organelle here, the chloroplasts, this one, leaf peroxisomes, and this one. This is mitochondria. But from the concept of cytology, these organelles, this is a double membrane organelle. A double membrane organelle, you see, and also this is a double membrane organelle with a folding like yes. Therefore, this is chloroplast, and this is leaf peroxisomes, and this is mitochondria. Okay, in chloroplasts, there is a carbon dioxide acceptor, rubip. This is 5 carbon compound. The rubip, once it accepts the oxygen, it forms the 2 molecules. One molecule with 3 carbon compound, or we call it PGA, 3 carbon compound, and another molecule of 2 carbon compound, we call it 2-phosphoglycolate. This is 2 carbon compound. Yes, as I told you, this phosphoglycolate cannot enter into a Calvin cycle. Rather, it is converted through different organelles from chloroplasts, peroxisomes, and mitochondria. But remember that this reaction is catalyzed by the enzymes known as rubisco. Okay, thereafter, after formation of this phosphoglycolate, this phosphoglycolate first is dephosphorylated in the chloroplast. It means the phosphate is lost to form the so-called glycolate. Yes, what next? This glycolate is transported into the leaf peroxisomes. Okay, this glycolate, remember that again, this is 2 carbon compound. You see, this is 2 carbon compound. The glycolate in the leaf peroxisomes is converted into another compound known as glycosylate. But the conversion of this glycolate into glycosylate, it uses oxygen, or the oxygen is using. Once oxygen it is used, it forms glycosylate and the hydrogen peroxide. If you remember from the concept of cytology, we have said in the peroxomes there is the so-called hydrolytic enzymes. These hydrolytic enzymes are able to convert this hydrogen peroxide into water and oxygen. Since this hydrogen peroxide is very toxic, therefore must be converted into another product, which is water and the oxygen. Again, this glycosylate is converted into another compound known as glycine. Again, this is 2-carbon compound, glycine. This glycine is in the mitochondria. This glycine in the mitochondria again is converted into another compound known as serine. Okay, but during conversion of glycine into serine, there is a carbon dioxide which is released, but also there is ammonia which is released. But again, in the mitochondria, the oxidizing node is converted into reduced NADH2. Ask yourself, from the concept of cytology, this glycine is 2-carbon compound, but this serine is a 3-carbon compound. How this conversion occur, 2-carbon compound to form 3-carbon compound? Actually, here, to make a 1 molecule of serine, we need 2 molecules of glycine to synthesize a 1 molecule of serine. Therefore, 2 molecules of glycine, 2 carbon, 2 carbon. Therefore, it has a 4 carbon. In this 4 carbon, 1 carbon is lost in form of carbon dioxide, and the remaining 3 carbon is the one which are used to form serine. Okay. This process we call it photorespiration because of what? The process, it uses oxygen. As you can see here, at the beginning, there is oxygen which is used. You see, again here, there is oxygen which is used. But again, here, there is carbon dioxide which is released. It uses oxygen, yes, but it releases carbon dioxide. Thereafter, this serine, after formation of this serine, is transported into the leaf peroxomes. Serine, 3-carbon compound. In the leaf peroxomes, serine is converted into another compound known as hydroxypyruvate. Again, this hydroxypyruvate is converted into another product known as glycerate. But again here, the products, the NADH2 is used to convert this hydrox into glycerate from NAD positive. Yes, now we have this glycerate. After formation of this glycerate in the peroxomes, this glycerate is transported to the chloroplasts. Therefore, now here we have this glycerate. Remember that this glycerate is a 3-carbon compound. After transportation of this glycerate from the peroxomes into the chloroplasts, this glycerate again is converted into another molecules of 3 carbon compound known as PGA. But conversion of this glycerate, it requires NH. Where does this NH come from? The NH comes from ATP. It means during this process, ATP is used again to produce ADP. It means inorganic phosphate is added to this compound so as to form the so-called phosphoglyceric acid. As I said, the process occurs under the presence of light, that is photorespiration. But it uses oxygen here, it uses oxygen, but also the process, it releases a carbon dioxide. One carbon is lost from glycine to form a carbon dioxide. The process, it uses ATP. You see here, the ATP, also ATP are used during this process of photorespiration. So, and that is why we are saying the photorespiration, it lowers the yield of C3 plant, because instead of this 2 carbon compound to enter into a Calvin cycle, it is exchanged between different organelles, and finally it is converted into PGA. This is photorespiration. Okay, as you have said, these plants are grouped according to the fastest stable product to be formed during photosynthesis. Therefore, we call this C4 plant because of the fastest stable product to be formed. Therefore, during photosynthesis, the fastest stable product has 4 carbon. Therefore, this 4 carbon compound which is formed during a C4 plant, it is known as oxaloacetic acid. Oxaloacetic acid, or we call it oxaloacetate. Okay, this is 4 carbon compound. But C4 plant, again, examples of C4 plant, because you need to know examples of C4 plant. Examples of C4 plant are sugarcane, millet, also sorghum, etc. Therefore, the way photosynthesis occurs in this is sugarcane, millet, and the sorghum is quite different from the photosynthesis of maize, soybeans, and so. Because of this, a fast stable product to be formed. This C4 plant, it involves 2 main biochemical pathways, and these are the first one is we call it hard slack pathway, and this pathway, it is because of these 2 scientists, the one which discovered how the photosynthesis in C4 plant occur, hard and slack. Therefore, this hard slack pathway, also known as a C4 pathway. How this C4 pathway occurs, we shall see this later. But also another pathway, as you said, there are 2 main pathways. The first one, hard slack pathway, but the second one is a normal pathway of C3 plant. We call it a Calvin cycle. Normal pathway C3 plant. Call it Calvin cycle. Therefore, C4 plant involve both Hatch-Slack pathway and a normal calving cycle in the previous lesson. Okay. What the difference between this pathway? The difference photosynthesis which occurs in C4 plant and C3 plant is because of the leaf anatomy of this plant. C4 plant, the leaf anatomes, it has two distinct types of cells, and these are mesophyll cells and the bundle sheath cells. Mesophills and the bundle sheath cells. In these two types of cells, each cell has its own chloroplast. The chloroplast of the mesophills has a grana. Therefore, here it is this type of cell is specialized for light-dependent reaction. But the chlorophyll of bundle sheath lack grana. And this is specialized for a dark reaction or light independent reaction. But also, in this C4 plant, there are different enzymes which are involved during this process. And these enzymes are rubib carboxylase and the pep carboxylase. There are two enzymes, rubib carboxylase and pep carboxylase. As you have said in C3 plant, rubib stands for ribulose biphosphate, but this pep stands for phosphoenol pyruvate. Pep stands for phosphoenol pyruvate. But again, these are enzymes which catalyze the reaction of C4 plant. The carbon dioxide acceptor of C4 plant are two. We have PEP, this is our carbon dioxide acceptor, and also we have this rubib. Therefore, fixation of carbon dioxide in C4 plant occur twice, because in mesophyll, again in the bundle sheath cells. This PEP, carbon dioxide acceptor, known as phosphoenol pyruvate. And you need to remember that this kind of photosynthesis, it is mostly for those plants which are found in tropical and subtropical areas. Okay. Therefore, in mesophils, as you have said, in mesophil cells we have a PEP carboxylase, but also we have PEP. In bundle sheath we have a RuBip carboxylase, but also we have a RuBip carbon dioxide acceptor, and also the enzymes. Okay, C4 plant, we have said, also we call it the Hatch-Slack pathway. Hatch-Slack pathway. What is this Hatch-Slack pathway? As we have said, these are two scientists, Hatch and Slack. Therefore, the pathway which was discovered by these two scientists is generally known as Hatch-Slack pathway. Therefore, when we are talking about this pathway, we are talking about the pathway which involves transport of carbon dioxide and hydrogens from mesophil cells to bundle sheath cells. A pathway which involves transport of carbon dioxide and hydrogens from mesophil cells to the bundle sheath. Bundle sheath cells. As you have said, this C4 plant has two different type of cell, mesophils and bundle. Therefore, the process or the cycle, the pathway which involves transport of this carbon dioxide and hydrogen from outer layer of cell, which is mesophils, to the inner layer of cell, which is bundle sheath, we call it Hatch-Slack pathway. This pathway, it involves four main stages, and these are the first stage, fixation of carbon dioxide in the mesophil cell. The second stage is mallet shunting. The third stage is regeneration of carbon dioxide acceptor called the phosphoenol pyruvate. PEP. This phosphoenol pyruvate fourth stage is refixation of carbon dioxide in the bundle sheath cells. Refixation of carbon dioxide in the bundle sheath cell. Therefore, these are four main stages of Hatch-Slack pathway. Okay, let's see how this stage occurs. First stage, as you said, is a fixation of carbon dioxide in the mesophil cells. Here, we have said the process occurs in the mesophil cell. Therefore, PEP, this is carbon dioxide acceptor in the mesophil cell, accept carbon dioxide. Once it accepts carbon dioxide, but remember that this PEP is a 3-carbon compound. When it accepts 1 molecule of carbon dioxide from the atmosphere, it forms a 4-carbon compound known as oxaloacetic acid. This is for a carbon compound. After formation of this 4-carbon compound or oxaloacetic acid, again this oxaloacetic acid is converted into another 4-carbon compound known as malate. Acid is converted into another compound, again is 4-carbon compound known as malate. But during conversion of this oxaloacetic acid into malate, there is a use of reducing power, one of the products of light reaction, which is a NADI pH2. It forms the oxidized NADI P. After formation of this 4-carbon compound, malate, again, this malate or shunting of malate or transport of malate from where? From mesophyll cells to bundle sheath cells. Therefore, what's happened here is a transport of malate from one cell into the next cells. Therefore, we have this malate. Malate is transported into the malate from mesophyll. Mesophylls is transported to the bundle sheath cells. Yes, after transportation of this malate, again, this malate in the bundle sheath cells, malate dissociate to form carbon dioxide, 3-carbon compound known as pyruvate or pyruvic acid. Malate dissociate form 3-carbon compound known as pyruvate and carbon dioxide Yes, once these products are formed, the carbon dioxide formed is accepted by another carbon dioxide acceptor in the bundle sheath cells. We call it RUBIP. But this pyruvate is transported back to the mesophyll cell so as to regenerate the so-called carbon dioxide acceptor, phosphoenol pyruvate. Yes, therefore, the third stage, as you have said, is regeneration of carbon dioxide acceptor. Carbon dioxide acceptor called a PEP. Formation of this pyruvate transported back. But during transportation of this or during regeneration of this, there is NH from ATP. ATP is used here to regenerate a phosphoenol pyruvate. The last stage is refixation. Refixation of carbon dioxide in the bundle sheath cell. It means we're talking about the refixation because the carbon dioxide already fixed in the outer layer of cell, which is mesophyll cell. Therefore, here refixation occur again, and that's why we are saying C4 plant, this fixation occurs twice, in mesophyll cell and in the bundle sheath cells. Okay, let me show you how this C4 photosynthesis occur. Okay, let's see C4 pathway, how it occur diagrammatically. First, we have a PEP. This is 3 carbon compound, a carbon dioxide acceptor. It accepts a carbon dioxide from the atmosphere, and it forms a 4 carbon compound known as oxaloacetate. After formation of this oxaloacetate is converted into malate. But during conversion, again, product of light reaction are used here, NADPH2, to form oxidized NADP. Malate, after being formed, again is transported into the bundle sheath cells. This is 4 carbon compound. Remember, this is 4 carbon compound. This malate in the bundle sheath cells dissociate to form a 3 carbon compound known as pyruvate. This is a 3 carbon compound, but also it forms carbon dioxide. Formation of this pyruvate, again, this pyruvate is transported back into the mesophyll cell, and again it is converted into a phosphoenol pyruvate, but with the help of ATP. But this process requires energy from 2 energy phosphate bond. Therefore, here we need 2 ATP, which will form 2 ADP. Once the carbon dioxide is formed in this part, this carbon dioxide is accepted. As you said, in this part we have a RuBip. You see, with the help of the enzymes known as Rubisco. Rubisco, carbon dioxide is accepted by this, and they form a 3 carbon compound known as PGA. See, after formation of this PGA, the process continues as a normal Calvin cycle. Normal Calvin cycle. As you see, carbon dioxide fixation is here. After this, there is a conversion of oxaloacetate to malate. Then malate is shunted. After being shunted, this malate dissociates to form carbon dioxide and also to form the pyruvate. But during this process, again, the malate, one of the products, oxidized NADP, is used to regenerate NADPH2. This NADPH2 is used in this normal Calvin cycle in the reduction stage. Okay, this is Hatch-Slack pathway which occur in C4 plant. Okay, to bring this concept clearly to you, let me take you to some of the sample questions that you may be asked from this part. A sample question number 1 is, briefly explain why the rate of photosynthesis in C3 plants is enhanced by higher concentration of atmospheric carbon dioxide, while in C4 plant it is not. Yes, up to now you know what is C3 plant, what is C4 plant. Actually, increase in carbon dioxide concentration in the atmosphere, it favors the rate of photosynthesis because it increases the efficiency with which the RuBip carboxylase can work on. When you increase carbon dioxide concentration, the rate of photosynthesis in C3 plants tends to increase. But in C4 plants, it is not enhanced by increase of carbon dioxide concentration. This is because the leaf anatomy of C4 plant. C4 plant has 2 types of chloroplasts, mesophyll chloroplast and bundle sheath chloroplast. the C4 plant has the ability to maintain the higher carbon dioxide concentration in the inner bundle sheath cells compared to that of the atmosphere. Therefore, even there is an increase in carbon dioxide concentration, cannot cause any effect on the rate of C4 plant, because of inner cells or inner chloroplast has higher concentration of carbon dioxide compared to that of atmosphere. Therefore, the C4 plant is able to maintain high carbon dioxide concentration in the inner bundle sheath cells compared to that in the atmosphere. See, this is a response we expect you to answer from this question. Yes, let me take you to another sample question, the question number 2, which read as, how the plants such as sugarcane and sorghum adapted to overcome the process of photorespiration. The response from this question, first you need to know, sugarcane and sorghum, are they C3 plant or C4 plant? And then after knowing this plant are C3 or C4, and then you can answer this question, how these plants are they adapted to the process of photorespiration. As I told you, these two plants, sorghum and sugarcane, these are C4 plants. Therefore, how this C4 plant adapted to overcome the process of photorespiration? As I have said, the C4 plant, from this diagram, you can see this diagram here, this is mesophyll cells and the bundle sheath cells. Therefore, the leaf anatomy of C4 of this sugarcane and sorghum, they have these two types of chloroplasts. And we have said in the mesophyll, the enzymes which is found there is only PEP carboxylase. And this PEP has no any affinity with oxygen. Therefore, it cannot fix oxygen. Therefore, it prevents photorespiration. But again, in these bundle sheath cells, there is a RuBip, is a carbon dioxide acceptor, but also there is these enzymes, Rubisco. As I told you, in the bundle sheath cells, there is no grana. The lack of this grana is an advantage, because the grana, it means the light reaction can take place, and this light reaction can produce oxygen. This oxygen can be accepted by this Rubisco. But for C4 plant, in the bundle sheath cells, there is no grana. Therefore, this is an advantage because it prevents photorespiration to occur. Yes, this is what we expect you to explain from this question. The sample question number 5 reads, differentiate between C3 and C4 plants. Students, as I told you, in different subjects, we have different way of answering the question. In the biology subject, when you are given the question of differentiating between A and B, what you are supposed to do is to know the criteria for differentiating between the two. Therefore, differentiate between C3 plant and C4 plant. Up to now, you know C3 plant and C4 plant. What the difference between C3 plant and C4 plant? Therefore, first of all, you need to use different criteria. In the criterion number 1 that you may use, you may say fastest stable product. For C3 plant, you remember the fastest stable product to be formed has a 3 carbon. But for C4 plant, the fastest stable product to be formed has a 4 carbons. Criterion number 2, you may use leaf anatomy. Actually, in C3 plant, as you said, there is only one type of chloroplasts. But for C4 plant, there are 2 types of chloroplasts. There are so many criteria. Again, you may use the fixation of carbon dioxide. In C3 plant, the fixation of carbon dioxide occurs once. But in C4 plant, the fixation of carbon dioxide occurs twice, because there are 2 types of chloroplasts. But also, you may use the enzyme involved as another criterion. Enzyme involved for C3 plant, there is only the RuBip carboxylase, or we call it Rubisco. But for C4 plant, there is PEP carboxylase in mesophyll cells and RuBip carboxylase in the bundle sheath cells. Again, you may use another criteria of a representative species. we saw the different example of C3 plant, example C4 plant. Up to now, I think you know. Example C3 plants like grasses or bryophytes, trees, also the rice and many other. But for C4 plant, there is sugarcane, there is sorghum, you see, and so. Therefore, the representative species is another criterion. But again, you may use the efficiency of photosynthesis. In C4 plant, the photosynthesis, there is high efficient. In C4 plant is high efficient in photosynthesis. But the C3 plant, because there is a chance of photorespiration to occur, there is low efficient in photosynthesis. Therefore, there are many criteria that you may use to distinguish between C3 plant and C4 plant. Okay, let me conclude my presentation by saying we have seen the C3 plant and the C4 plant. Therefore, up to now, you know the examples of C3 plant, how photosynthesis in C3 plant occurs, how photosynthesis in C4 plant occurs. You see, up to now, you can explain the Hatch-Slack pathway. You see, you can explain the Calvin cycle, and so. Thank you so much for listening. Let me end up my presentation today. Join me on the next session, which we shall talk about the issue of a factor affecting rate of photosynthesis. Thank you so much. Bye-bye.
Kipindi cha Darasa Online kimeletwa kwenu na Chuo Kikuu cha Sauti Mbeya.
Mimi naitwa Ali Rajabu mwenye mvua. Ni msanii mchoraji wa picha za wanyama na nyingine tofauti tofauti. Tunaanza kuchora ni mbingu kama unavyoona blue. Na baadhi ya picha kwenye mbingu tunaweza kuweka hata mlima Kilimanjaro ili kutangaza pia nchi. Tunapatia hii ardhi, watalii wanapotembelea nchini kwetu wanaenjoy sana. Ukiona picha tofauti tofauti zimechorwa za wanyama, za Masai na kuangalia mazingira waliotembelea kwa mbugani unayakuta yakiwa picha.